Okay, this is a video lecture for Chem 255, which is Introduction to Biochemistry at St. FX University. When we when we left class last time, we were on this slide. We had finished discussing how tRNAs are shaped, what their role is, and now we're putting them into practice. So we know that tRNAs have to bind messenger RNA. We can see over here on the right, messenger RNA is the transcribed message from DNA, which are all the instructions to code for proteins in the body. And tRNAs are these cloverleaf structures here, which attach or recognize three codon sequences, or three nucleotide sequences in what's called a codon. Now, the specific codon finds or targets a specific amino acid, that is, a tRNA carrying a specific amino acid. And these amino acids are linked together to form proteins. And so you can see how the sequence of nucleotides in mRNA would influence the structure of protein. So there are two jobs of tRNA. One has to bind the right codon. Two, it has to carry or transfer the right amino acid. Now, we're not going to focus too much on how tRNA is charged with the right amino acid. That's a bit outside the scope of this course, but we will talk a little bit about how tRNA recognizes the code and what the code means. So the code, like I said, references specific amino acids. It's degenerate, which means that it is redundant. We'll talk about that in a second. It's non-random, so there is a pattern to it. There is a reason for the sequences in the code. And it's pretty much universal. There are some exceptions where the code is not universal. Some mitochondrial DNA uses a different sequence. Some prokaryotes use a different sequence. But for our purposes, the code, the genetic code, is universal. Nearly all organisms use the same genetic code. So we just take for granted that it's a triplet code, that there are three bases that comprise one codon. It can't be a single base code. There are 20 amino acids that we know of, and only 4 nucleotides. So we could only code for 4 amino acids if it were a single code. Single obviously can't work. Doublet code also can't work. You only have 4 options in each of the 2 spots, and so you can code for 16 amino acids. So the triplet code is the first one that would work. But with 4 options, 4 nucleotides possible in each of the 3 spaces, we can have 64 possible codons. So there seems to be a bit of overkill here. Now, in this picture, I want to point out that we always translate mRNA from the 5' to the 3' end. So this tRNA carrying arginine would bind first. This tRNA carrying glycine would bind second, and alanine third. And the amino acids are added in the order that they bind mRNA. So from the 5' end to the 3' end. Now RNA itself, like I said, binds from 5' to 3', and it requires, it necessarily needs ribosomes to carry this out, to hold the messenger RNA and then to bind the transfer RNAs in the appropriate spots. So the ribosome is the complex that puts all of these things together, and it itself is made of RNA, which is important for a point that we'll make later. Now, at any one time, 3 amino acids bind the ribosome, and there are 3 binding sites related to the job of that tRNA. So the first one is the exit site, or the E site. This is a tRNA would be bound here that has already transferred its amino acids. The middle site is the peptidyl site, peptidyl tRNA site, where the chain, the evolving protein, the elongating protein, is currently held. And then the third site is the site where incoming amino acids bind, and this is the aminoacyl tRNA site. So this is a staging area where the next amino acid bound to its respective tRNA would be in place in order to donate or add its amino acid to the evolving chain. We can see these three sites here. Each of these tRNAs is labeled for its position on the mRNA. The far left is the aminoacyl site, peptidyl site, and then the exit site. And you can see on the exit tRNA, it doesn't have an amino acid. It's released its amino acid. The amino acids are these globular looking proteins here, and so it's now ready to not be discarded, but detach from the mRNA and recycle or have an amino acid re-added to be used later. The peptidyl site is the one that carries the amino acid chain, and you can see here the amino acid in the aminoacyl tRNA site is being added to the peptide. So when you make a protein, the aminoacyl tRNA is added at the proximal end of the protein. That is, the polypeptide is added on top of the aminoacyl tRNA. So right now what we're looking at is the peptidyl tRNA site is actually transferring the protein that is currently attached to it on top of the amino acid that's attached to the tRNA at site A. And so in that sense, making a protein is a lot like casting a fishing reel. It extends from the reel. And here the ribosome is the reel. This is where the amino acids are added at the base, the proximal end of the protein. So, given what we know about mRNA, a natural question is, which is the 3' end and which is the 5' end? We said that mRNA is always transcribed from 5' to 3'. So take a minute just to think about this. Pause the video if you have to, and try to figure out which end of the mRNA chain here, either this one on the left or this one on the right, is the 5' and the 3' end. Okay, the answer, I'm assuming that you've all paused the video to think about it. The answer is that the 3' end is here, the 5' end is here. We translate from this end to this end. So a tRNA that has released its amino acid and is in the E site is closest to the 5' end. This would have been the first one to bind. It's already held the protein. It's already transferred it to the aminoacyl tRNA next to it. Now you can see the whole complex in this figure here. So this is zoomed out a little bit. We have the mRNA that we're used to. These three colored triangular globs are the tRNAs. Here we have the A site, the P site, and the E site tRNAs. And then this yellow and blue complex are parts of the ribosome, the things that hold them together. So you can see the machinery here, how it holds or it positions the mRNA to be accessible by tRNA. And then the amino acids that are being, the polypeptide that's being elongated by adding amino acids is this long yellow thing here. So this is where the polypeptide exits the ribosome. New amino acids are added here, and this just gets progressively longer and longer. And so think about the same thing as on the last slide. Which would be the 3' end versus the 5' end, number 1 or number 2? You can pause the video if you'd like, but if we hold in our minds the rule that the E site tRNA would have been the first one to bind and release its amino acid or add it to the polypeptide, and now it's leaving, the brown end here would be the 5' end because it's closest to the E site tRNA. Now, if you guys are interested, this video here, I'm not going to show it, gives us a great idea of how this happens in real time. So we can see the process of polypeptide elongation and how tRNAs fit in with the ribosome. So I'll include the link to this video in the description. Notice, before you watch that, notice as you're watching the shape of the mRNA, what it looks like in the cell. Notice the cellular locations too, how everything is set up, the relationship between the ribosome, the mRNA, the tRNA, and then look at the protein shape too as it's released. what happens to the protein as it is uh, gradually elongated. Now, at the end of the video, if you've watched the video now, it's pretty amazing to think that this is the way that all proteins are built. Hemoglobin in the video is one example, but muscle proteins, any of the enzymes that catalyze pathway reactions, or collagen in bone, any protein in the body is made this way. We take it for granted that the mRNA will encode for a protein. It's the right length to make a protein. It has the code already in place. But how do we know that the mRNA of the right length is produced? We already talked a little bit about how zDNA generally signals the start of a gene. But aside from that, how do we know that the mRNA is appropriate and will code for an amino acid? It seems that we do this through the use of what are called promoters. They specify where the machinery that binds to DNA is supposed to bind in order for it to access the start of the gene. So in this picture here we have a bunch of different genes. These are all individual genes that are stacked on top of each other. You can see the DNA code here. We know it's DNA because it includes thymine and it doesn't include uracil. And the promoter is a region that is roughly 40 base pairs, contiguous 40 base pairs from the start of the gene. So the gene starts at the initiation site, and then around 40 base pairs or so before that is what we consider the promoter region. And there are specifically two regions that are highly conserved. These are in E. coli, so they may or may not be applicable to humans. But in general, these blue shaded and the orangey peach shaded regions are highly conserved, which means they're probably pretty important in initiating the transcription of a gene. Now, not all of these sequences are identical. Remember, random mutations can happen. But if we look across a number of genes and a number of organisms, we can get what's called a consensus sequence. This is the average base pair in this position of the blue shaded region. Each of these numbers below each one says this is the percent that this nucleotide occurs. Same thing with this peach colored area here. The consensus sequence is TAT-AAT. The initiation site, on the other hand, can have pretty much any nucleotide in place. That will really depend on the first amino acid in that protein to be translated. So a bit more variability here. This consensus sequence seems to give us an idea of what the promoter should, what the code that the promoter should have. Now, all of these sequences do vary considerably. If you look at the individual sequences here, you'll find that they're not all TAT-AAT in the case of the orange, or the same thing in the case of the blue shaded region. This doesn't mean that it won't work as a promoter. If the code is different, it just means that the effectiveness of this promoter region is different. So it affects what we might call the initiation efficiency, the ability to turn on a gene in response to a given stimulus. So this is really interesting. This could be one of the factors that relates to genetic predisposition. Some people seem to have a high metabolism or respond to fitness programs quicker or respond to hot and cold environments better than some other people. And it could be, in part, that the genes encoding proteins that are relative to those situations, or relevant, not relative, the genes encoding those proteins are turned on more quickly or more slowly, depending on the initiation frequency, the initiation efficiency of the promoter. Now, it is imperative that we have some way to anchor the machinery at the start of a gene. It's imperative to have a promoter region that is intact and functioning to make a proper protein. And the reason for that is there's no way to know where to start, one. We could look at the wrong place in a chromosome. But we also have to really fine-tune where in that region we start transcribing the protein. We could have 3 different frames of reference in the same section. So without any anchor, we could have the first reading frame, which codes for tyrosine. If we didn't have a promoter that anchored the machinery and made mRNA that started at the right nucleotide, we could also have a second, completely different reading frame, where in this case, instead of tyrosine, we get threonine. And the same thing goes for the third reading frame. This code CUA translates into glucine. So the promoter really serves to anchor where the mRNA starts. And then where the mRNA starts will influence which amino acid is the first amino acid in that protein. Now we know that the code is sequential. That is, one 3-letter codon means one amino acid, and then we start at the subsequent 3-letter codon for the second amino acid, and so on. It was thought for the longest time that the codes overlapped. That is to say that this UAC would code one amino acid, and then we might move and code ACU next, and then CUA next, in an overlapping fashion, which really would be a great way to economize, right? If we could store so much information and overlap the genetic code, we could store either a lot more information, or we could compress all of our DNA down into a much smaller package. Overall, an increase in economy. But this presents some serious problems when we think about it a little bit. And it's in relation to the force that drives selection and evolution. We know that single amino acids, and sometimes whole chunks of amino acids, but single amino acids can change. They might be copied improperly when we replicate DNA, and that will affect the mRNA that is transcribed from that DNA. Now, it's not a problem in practice for us as we live now, but if the code were to overlap, and say, for instance, this C turned into a G, you wouldn't affect only this first amino acid, but you'd affect the second one and the third one, because this nucleotide is used in all 3 reading frames. And so all of a sudden, these random chance genetic mutations, if you will, cause 3 different amino acids to code differently. 3 amino acids that wouldn't have been in that protein are now there. And the sequence of amino acids, the characteristics of that protein, and the shape, ultimately the function, will be affected. So it really is a benefit that we use this sequential coding to code for amino acids. However, the code being degenerate allows a certain amount of redundancy. We don't want even our sequential code to change the amino acid that will be inserted with one small substitution. So there is a lot of redundancy in the code, and this is partially explained by the wobble hypothesis. So you can see here that the anticodons, which are the regions of the tRNA that recognize regions of the mRNA to be transcribed, or that indicate a specific protein, these have a bit of play in specifically the 5' nucleotide. Here, this GM is methylated guanine. It's not the same as the guanine base that we include in DNA and RNA. It has an extra methyl group attached. And what that allows it to do is bind not only to cytosine, but also to uridine. And so we have a bit of play in this third position of the code, where the same tRNA will bind to 2 completely different codons. So if this changes by random chance, it's likely, or I guess the proper term is to say, it's less likely that the sequence of proteins and the function of that protein will change. More specifically, the sequence of amino acids won't change, and the overall shape and function of the protein won't change. So it doesn't follow strict binding. Both of these here will code for phenylalanine. Now we can see that this isn't the only place where there is redundancy. If we look at the entire genetic code as it's laid out here, and we don't have to memorize these, we probably won't use a lot of these individual codes, but the principle is what we're looking at here. On the left we have the nucleotide in the first position of the code. On top is the second position, and then on the right, these 4 rows within each larger box indicate a different nucleotide in the third position of the codon. And for the most part, there's a fair bit of redundancy. Consider leucine. It doesn't matter what the third nucleotide is, as long as the first 2 are Cu, the third one can be anything. And that region will code for leucine in an mRNA. So it's degenerate. There's a lot of redundancy. Most amino acids will recognize, or the tRNAs bound to most amino acids, will recognize multiple codons. Alanine, valine, threonine. Few exceptions. Methionine only recognizes one. There's a stop codon that tells the ribosome to stop. These are recognized by singular codes. Now what's really interesting, and we don't have to go into a lot of detail about this, but it seems very random. It seems random. Why pick a C here and a U here and a G there? It seems very random, but in practice it's not. There seems to be a code within how the codes are laid out, a code within the codons. It seems as though this first position here, seems to relate to specific amino acid precursors. So in the body, we can make amino acids from other metabolites, things in metabolism that are part of different reactions. We can use those to make amino acids. And more often than not, this first letter will correlate, will identify that precursor. Some things, we don't know them yet, but we'll get to know them. Some things like pyruvate or alanine or alpha-ketoglutarate. And we'll see these a lot in the TCA cycle. This is one area where I will highlight this. These precursors can be turned into amino acids. And so there's some relationship between the code here and a precursor that will generate a specific amino acid. The second position identifies a transformation. You can almost think of this as instructions. How would you change that precursor to obtain that specific amino acid? The second letter seems to be a transformation. So add a methyl group, or take out an amine group, or add an amine group. The second position identifies what happens, and that lends a specific characteristic to an amino acid. Generally, the second position will relate to how hydrophobic an amino acid is. And so you can start to see glimmers of how if we substitute maybe the second nucleotide, it might change how hydrophobic that amino acid is, but overall it doesn't seem to change it very much. It might be slightly more or slightly less, but the characteristic of that point in the polypeptide chain will still be hydrophobic. So hopefully the folding of that protein and the structure, the function, won't change very much. The third position for our purposes seems to be fairly redundant. There's little significance to what nucleotide is in that third position. It's not no significance, but little significance. The overall effect of all of this is to preserve the shape and the function of proteins. So if we mutate one area and we substitute a similar amino acid, we accomplish that goal. Now, seeing this little bit of a pattern to the codons has implications for how this whole system started. It's really a chicken and an egg problem. We have DNA, we have RNA, we have protein. Protein helps to process DNA. RNA helps to process RNA. We're not sure which one came first, but the code within the codons might give us an idea of which one came first, and it's called the RNA world hypothesis. It aims to help explain this chicken and the egg problem. So we've noticed, first of all, that RNA strands themselves are catalytic. They do work in the cell. A ribosome is RNA. A tRNA is RNA. So if strands are catalytic, if they can take on these different shapes and catalyze reactions, why can't fragments do it? Why can't a dinucleotide or a diribonucleotide, why can't that do a job in the cell too? We've noticed that there seems to be a lot of machinery, RNA machinery, for processing DNA and protein. There isn't so much machinery to process RNA. On top of that, the critical components in the cell are RNA, or at least based on RNA. We showed how NAD, FAD, ATP, and CoA all have roots based on adenosine, the ribonucleotide adenosine. And so it seems that RNA is integrated into a lot of key functions in the cell. Not only that, but we'll see in an example in a bit how DNA, we won't see this specifically, but we'll see an example of how nucleotides are synthesized. And when we make DNA, we synthesize it from RNA. We don't make it from scratch. We synthesize it from RNA. RNA is made first. So this implies something to the sequence of how these might have arisen. Now, if we speculate, the theory goes that sometime way in the distant past, when we had nucleotides, we had pyruvate and some key elements, which are very simple molecules. It's possible that a couple RNA molecules got together. Think about two ribonucleotides getting together. This is possibly a catalytic RNA fragment. Now these two little ribonucleotides might have ran into other things that were in proximity. What if these bound? Maybe it's specific, the binding was specific to which two ribonucleotides you had bound together. And so it's possible here that we see why one of the RNAs might have dictated which precursor would have been bound, and why another ribonucleotide might have dictated what would happen to that precursor. And so now we start to see how these little fragments might have been the basis for a code. So what if all this happens? What does that mean? Well, if this happens, the end result is that you have an amino acid bound to a small length of RNA. You have an amino acid bound to RNA. This could be the roots of tRNAs. Who knows? Then, if you have this little triplet, this amino acid and two ribonucleotides that are now happily moving along, wherever they're moving along, if they run into a bigger RNA molecule, then just for the virtue of that molecule being bigger, the amino acid would transfer. If those amino acids get transferred over, if they get linked together, if that does something, if it's beneficial, then all of a sudden this becomes important. Maybe we have the basis for RNA dictating sequences of amino acids in a protein. This is wild speculation. It's not possible to know this. We don't have experimental data to back this up yet, but it's a pretty elegant theory, I think. The biggest question left after this is, well, there's only two ribonucleotides here. The code is a triplet code. So where and how do we make the jump to a triplet code? And again, mostly speculation. We don't know for sure, but if the ability of the diribonucleotides to fit onto a larger RNA molecule, if that was affected by having a third ribonucleotide, then it's not too hard to imagine that a third would be incorporated. Maybe it was close quarters, and then having a fairly large amino acid bound to two ribonucleotides, maybe it wouldn't let two of them fit side by side. But if you added a third, maybe it would give the amino acids a bit more room. Who's to say for sure? We don't actually know. Now, the major assumptions here are that we had ribonucleotides. We don't know how those are created yet. And we also assume that we had pyruvate, alanine, alpha-ketoglutarate, a bunch of basic molecules. So these are pretty important assumptions, and it sounds like they're a little bit ridiculous. How can these things already exist? Well, it might not be as impossible as it sounds. And I'm going to show you an example of how ribonucleotides are made in the body currently. Then I'm going to propose a way that they might have been made earlier, not in the body. So let's take pyrimidines, for instance. This is going to be an example of how uracil is currently made in the body. The pyrimidine ring simply takes aspartate. This big chunk comes from aspartate, the amino group from glutamine, and then a carbon from bicarbonate. These are the three main ingredients for pyrimidines. Then we modify them slightly. We need some energy, and we need a ribose to make a nucleotide. Well, specifically a nucleoside. We need both a ribose and a phosphate to make a nucleotide. But it's relatively simple to make these, not a lot of ingredients. So, in the body, as we know it right now, as is happening right now, we can make uridine monophosphate from scratch, just with bicarb, glutamine, which donates the nitrogen group, water, and energy. That's all we need to start. First step is that we have to add nitrogen and a phosphate to essentially the bicarb backbone, if you will. We get phosphate from ATP, we get the nitrogen from glutamine, and now we have this molecule called carbamoyl phosphate. This is the starting point for nucleotide synthesis. Now, the other main ingredient on the last slide was aspartate. Aspartate is still a fairly simple compound. It has this characteristic of looping back around once it's attached to this carbon from bicarbonate. And already you can see the characteristic nature of the pyrimidine ring starting to form, just by the nature of the shape of aspartate when it binds. And so, if these two things happen, it's really easy to make a pyrimidine ring. All you have to do is link the nitrogen to that carbon here, and as it turns out, it's quite easy to do. All you have to do is dehydrate. We dehydrate, which links this in a pyrimidine ring, and then we oxidize. So we remove electrons from the pyrimidine ring, and now we have what is considered a pyrimidine precursor, ororate. Now we have this pyrimidine precursor, and we need to attach a ribose and a phosphate. In the body right now, we use this molecule, PRPP, which is phosphoribosal pyrophosphate, which is a mouthful. This simply says phosphate bound to ribose and bound to pyrophosphate. You can pretty much think of this as ribose triphosphate. Now, this is a process that requires a fair bit of energy, binding this ribose phosphate to ororate. And luckily, breakdown of pyrophosphate is very exergonic. It's likely to happen spontaneously. So we get energy from breaking this phosphate bond, and then we get energy because pyrophosphate pretty much immediately dissociates. What we're left with is this pyrimidine precursor ororate bound to ribose and a phosphate. How many steps are left? Two things, or one thing, sorry. We just have to decarboxylate. This carboxyl group here is removed. We're left with uridine monophosphate. We're left with a nucleotide. So all we have to do is cobble together bicarbonate, amine group from glutamine, a carbon ring from aspartate. Then we have to join on a ribose, we have to dehydrate and oxidize it, and also decarboxylate it. But these are all things that could have happened, possibly, on their own before we had enzymes. And this is a really cool experiment that I'm going to just illustrate for you guys briefly that looked at this. Here, the whole goal was to simulate the energy that would have been released from an asteroid impact. They're using lasers to do that here. You can see the setup with the laser beam that is focused through a lens and reacts with a sample. This is just a basic carbon-based molecule sample called formamide. This is thought to be a pretty likely precursor to a lot of organic compounds that would have been around on prehistoric Earth. And you can see how this is set up in this lab here. This is the laser. Here's the sample. And we deliver energy to the sample to see what happens. Well, what happens? If we take a lot of energy in the form of a laser and we apply it to formamide, the products of this reaction react with itself to form these other more complex molecules. What's really interesting is that these reactions continue. Once these radical products are in place, they are highly reactive and they react with the formamide derivatives that are in the solution. And it's been documented, this study documented the formation and the process by which we form every single ribonucleotide. Adenine, guanine, cytosine, and uracil. Or rather, every single nucleotide base. But every single base forms spontaneously when we apply this massive amount of energy that is analogous to an asteroid impact to formamide, which has radical implications for how we think this happened. If these nucleotide bases, these are RNA bases, were formed through this high energy transfer from something, we would have them floating around in solution. They would be free to react with each other and any other products that were around. All of a sudden, the RNA world hypothesis does not seem so far-fetched. It's still quite far-fetched, but it's not so far-fetched. And so I want to leave it there for now. This will be the end of our DNA lecture. We're going to move on to proteins and amino acids in our next class. If you have any questions about this, please email me or comment here even. Ask on Top Hat, and I'll be happy to help you out.